Okay, thank you very, very much. I want to thank the organizers in particular for inviting me to come here today. I want to describe single molecules, but I have the pleasure of doing it in a spectacular room, and I'm very, very happy to do that. And really in specific, I want to talk about how imaging and photo control of single molecules leads to super resolution by a different method than that described by Stefan Hell. So, but to do this, we have to go back into the mid-80s for early inspirations where people were looking at single molecules by STM. They also were seeing single ions in traps. And I was asking the question at that time, could we do this with a molecule? Can we see a molecule in a solid, for example? Why not? Well, unfortunately, there were some people who thought that this was pretty difficult. The great physicist Erwin Schrödinger, who is of course, one of the founders of quantum mechanics made this statement back in 1952, the year before I was born. But I didn't much know about it at the time. But never mind what his particular point of view was, there were also lots of other scientists who felt that it was going to be impossible to see a single molecule. So the real part of the story is, how did we come to think that it was possible? And in order to describe that to you, I have to go back and tell you a little bit about some fundamental spectroscopy to get everyone on the same page in this broad audience. Maybe we could adjust the projector so that you could see the top and the bottom of the screen. This is showing an absorption spectrum. That is, absorption is a function of wavelength for this molecule, pteryline, in a host crystal of paraterphenyl, which is transparent. It's the pteryline that's absorbing here, this conjugated molecule. At very long wavelengths, there's no absorption. And as you go to shorter and shorter wavelengths, higher and higher energy, you finally turn on electronic transitions and then some additional vibrational transitions. In these plots, I'm showing wavelength increasing to the right, and here's a color scale to orient you with our visible light. But if you remember that frequency and wavelength are inversely related, then frequency would be increasing to the left. And the frequencies on this plot are really huge. 460 terahertz, 460 times 10 to the 12th counts per second, cycles per second, and here 860 terahertz. So now I want you to go to low temperatures with me. We're going to watch this transition, the first electronic transition, as we cool to low temperatures. And in order to then keep track of everything, we're going to have to expand the scale by a factor of 25 times, so right around this sort of orange region. Here's what you see. Here's the orange now, but now the color variations are hard for you to see. But high-resolution spectroscopy allows you to get great detail. Now I've switched the direction so that frequency is increasing to the right and wavelength to the left. And this is now a smaller range of frequencies. This transition is the first electronic transition, and you see it's become very, very narrow. That's the point here. Cooling to low temperatures, these molecular absorptions get extremely narrow. In fact, so narrow that you can now see that there's 4 inequivalent sites inside that crystal. But we have to keep going further. Let's now realize that this isn't even a resolved spectrum of this first transition. It's much, much narrower, so we have to increase expand the scale a whole lot more by another factor of 1,000 to see a bit more about what's really going on. So here's a schematic of what happens after that expansion. Now I'm only showing you 5 gigahertz with the center of this being 506 terahertz. And what you see here is that the individual molecules have become so narrow in their absorption that there is a distribution of where they are located in frequency space. That's because their different local environments have slightly different strains and stresses. And those very small effects work here because the lines themselves are so narrow and so sensitive to those perturbations. This produces a line that we call inhomogeneously broadened. And it was studied by many scientists all over the world around those times. In particular, an interesting application was envisioned. You want to really observe these individual widths. You would like to observe them, but you have this broad line profile giving you trouble. And so people would use photon echoes to see the homogeneous width, it's called, or something called spectral hole burning. And the way spectral hole burning works is you think of this inhomogeneously broadened line, but now bring your laser into resonance with only a small subset. Of course, the laser is much narrower yet, and so you only pump those that are in resonance. If you can change them so that they go away to some other place in frequency space, then a dip is left behind, or a spectral hole. That's what we mean by a spectral hole, that dip. And you can choose to make these holes at any wavelength you like in this inhomogeneously broadened line. In fact, the width of the whole line compared to the compared to the homogeneous width is about a factor of 1,000. So you could write many, many different bits at different colors. Now you see the connection to IBM. We wanted to use this as a frequency domain optical storage scheme. Writing bits, zeros and ones, at different wavelengths or different frequencies, and that's what allow you to have very, very high density optical storage. And a number of scientists were exploring this at IBM and other places at those days where I was working. Well, the nice thing about being at this great industrial research lab at that time is that you could also ask deep scientific questions, fundamental questions about this potential application. And the question that I was asking, what are the ultimate limits to this frequency domain optical storage idea? What would be the ultimate sort of smallest features that you could observe? What would prevent you from seeing small features and all of that sort of thing? And it turns out that the fundamental question that was important for our talk today is that we could ask, is there a spectral noise or some sort of spectral fluctuation that results from number effects or statistical number fluctuations or the discreteness of individual molecules, which would influence the smallest hole that you could detect. So to explain this a little bit more, I have a little simulation here. So think now of just a probability experiment. We have 10 boxes, and we're going to throw 50 balls at those boxes, where all boxes are equally likely to capture a ball. So you might think, well, maybe I'll see this after one experiment, where 5 are in every bin. Okay? But you know that's not what's happening, of course. This is a probability experiment. And in reality, what you're going to get is different numbers of balls in the different boxes. So while there might be an average of 5, there is a fluctuation about the average that corresponds to what we call number fluctuations. And the size of this variation, okay, around 5, is the square root of 5, the square root of n. This is the square root of n scaling, well known in statistics and probability, that arises from sampling. Now, you want to think of this axis here as the frequency axis that I just talked about in spectroscopy. And we've spread out the inhomogeneous line, so it's totally flat here for us. And what this is all pointing out for you is that there ought to be some spectral changes as you move from wavelength to wavelength coming from this effect, if you are sensitive to the fact that there are small numbers of molecules or there's discreteness in the number of molecules. Okay, so that's the idea, and here's a simulation just to prove it. Let's just throw molecules at frequency space, but do it with a Gaussian probability distribution. At low numbers, you see what you could get in one realization. As you put in more and more molecules, it seems to get smoother and smoother, but of course it never really smooths out. It always has a fluctuation that has the amplitude of about the square root of the number of molecules in resonance. The key thing is, this had not been observed in 1987, 86, 85. It had not been observed ever before. And so we first set out to detect this effect. With Tom Carter, we looked at penacene molecules as the absorber in a paraterphenyl host crystal, and what do we see was this. This is a piece of that inhomogeneously broadened line over a fairly small frequency range where the center of it is 506 terahertz. Let's blow up a piece of it and you see this unbelievable fine structure. And that's totally repeatable. If you scan it again, you see that fine structure again. Remember, we're at low temperatures here. So that is statistical fine structure. Now, how does it relate to our problem of detecting single molecules? Well, remember that this size of this effect, the amplitude of this effect, scales as a square root of the number of molecules in resonance. So suppose you had only about, let's say, 1,000 molecules contributing at every wavelength, then the square root of 1,000 gives the size of this effect, which is about 32. The point here is that you only then, once you've seen this effect, you have to work only 32 times harder to get to this single molecule limit of m equals to 1. We're already seeing an effect that comes directly from the discreteness of the individual molecules. We achieved this measurement by doing something called FM spectroscopy, which measures only the deviations of the absorption from its average value. So to us at that time, to me and my postdocs, it seemed perfectly reasonable to push this method that already detected fine structure for the first time to the n equal to 1 limit, because it's only 32 times harder. So the single molecule limit is within reach. So the next postdoc, Lothar Kador, and I actually did this, again for pinnacine and paraterphenyl in 1989, 25 years ago. The absorption of a single molecule may be viewed as a little line like that. FM makes 2 copies of it, and we used an additional modulation to remove some technical background signals. So a single molecule signal looks like this little W, and you can see it here. Here's where no molecules are present, and so on. This was done by FM spectroscopy, as I said, something invented by Gary Bjorklund back in 1980, and something I learned at IBM from him. Here's how this works very briefly. A laser that has no modulation is modulated by phase. Its phase is modulated, or frequency is modulated, as you see here. And the point of the whole experiment is that the sample, which is here, disturbs the balance between these 2 sidebands, so that this beam, which has no amplitude modulation, becomes an amplitude modulated beam, and you detect that amplitude modulation. The method is insensitive to scattering. It's limited by laser shot noise. We worked at the quantum limit, and there was actually a great challenge in doing these experiments, because actually this is the hard way to detect single molecules. That's because your laser intensity cannot be made really large to reduce the relative size of laser shot noise. We had to keep from broadening the molecular absorptions, which would make the signals weaker. So we had to work at fairly low intensity to do this. But it certainly represented a proof of principle. This business of using essentially FM radio at 506 terahertz to detect a single molecule. Well, a very important step occurred a year later. Michel Airy in France used the same system and proved that he could detect the single molecule absorption, but now by detecting the emitted fluorescence. Okay? And so he didn't use FM spectroscopy. He simply scanned the laser and measured the fluorescence emitted from the molecule. Here's the inhomogeneously broadened lines, one of them and another one, and these dots here are the single molecules. So this experiment, however, is sensitive to scattering from the sample. So the surfaces of the sample have to be very smooth and not produce any scattered light, which could affect your ability to detect these weak emissions from the single molecules. Rayleigh scattering and Raman scattering produce potential backgrounds, but overall, you can do that. You can make those clear samples. So this method produced higher signal-to-noise ratio for equal bandwidth. And so all of us switched to this fluorescence method after this. So here's some examples of what you see when you use that fluorescence excitation method. This is Pat Ambrose in my lab. Here's the full inhomogeneously broadened line, again at low temperatures for pinnacine and paraterphenyl. Blow it up, you see single little peaks. Blow it up again. You see this beautiful line from a single molecule that is only 7.6 megahertz in width at a center frequency of 506 terahertz. It's incredibly high resolution. 50 million times is the ratio between the center frequency and the width of these features. So they were beautiful for spectroscopy. You could perturb them with Stark effects. You could do so many interesting experiments that occurred in the early 90s. But we also were doing imaging too. We measured this image very early on where we scan the laser on this axis and we move the laser spot across the sample on this axis. So now what's happening in the spatial domain is really using the single molecule to directly measure the intensity profile of the focused laser spot. Now the focused laser spot has one of these intensity profiles limited by diffraction. So this is interesting because it connects back to what we have to think about next. And later, Urs Wildt's lab showed you can do this with wide field, two-dimensional microscopy in reverse contrast, and so on. Well, as soon as you get to a new regime, you see surprises. So here's some spectra on that old oscilloscope, 400 megahertz wide, and look what's going on. The single molecule at liquid helium temperatures in a crystal is jumping around, jumping from one frequency to another. When Pat and I saw this, we really jumped out of our gourd. We said, what's going on? This is really amazing. And it turns out this is a reflection of spectral diffusion at low temperatures coming from changes in the nearby host dynamics, which was then treated theoretically by Jim Skinner. But you can see there that it's a beautiful, and look at what we're doing. We're just taking a camera and putting it in front of this old oscilloscope and taking just sort of a movie to show you this. Very, very low tech back in those days. What's another surprise? Thomas Bachet looked at perilene in polyethylene, another molecule, and here you see perilene, a single perilene molecule being scanned by the spectral scanning, of course, and nothing's changing. But then if you bring the laser into resonance with it, it'll jump to another wavelength, and so the absorption is gone when you measure the spectrum again. Wait a little while, the molecule comes back, and you can push it away again. So we could drive the molecules back and forth, even at that early time. So there's both, if you like, jumping, or this would turn into blinking, by the way. If I put the laser at a fixed frequency in this spectrum, then the molecule is in resonance and out of resonance, so it blinks on and off and on and off. But here you can also control it. Well, at this time, since single molecules have been detected, I just want to remind everyone of the reasons why this is fascinating for a lot of interesting scientific reasons. We're removing ensemble averaging. We're not just averaging over billions and quadrillions of molecules and just getting one number, but we're measuring each one and measuring something about each one. That allows us to explore heterogeneity. Are these different molecules the same, or are they behaving? And of course, I showed you dramatic heterogeneity right there. You can follow state changes in time and test theoretical models of the system under study. You can also image and detect nanometer scale interactions. So a number of people started using FRET and other methods. And these days, I'm also just mentioning that there are commercial applications of being able to see single molecules by sequencing DNA and super resolution microscopy. But to describe those things, we have to get out of the low temperature regime and jump to room temperature. So a parallel thread that was going on in those days at room temperature, since you can't quite read this, the important thing now is room temperature. There were a number of interesting milestones that started way back in the 70s when people were doing fluorescence correlation spectroscopy. That's a situation where you just fix the laser beam in the sample and molecules diffuse through it. And whenever they diffuse through, you see a burst of light. So you can measure an autocorrelation function, even at concentrations as low as 1 fluorophore in there at a time. That takes a long time to accumulate that autocorrelation function. But later, single bursts, or actually the same year, 1990, single bursts were detected from a molecule going through the focus. Later, imaging of a single fluorophore was demonstrated with near-field methods that you'll hear about from Eric in a moment. And then confocal and wide-field imaging, all of these were done to collect the light from the individual molecules, either, let's say, in vitro or even in cells, to see single lipids moving on the surface of a cell. So many advances started occurring when we moved to room temperature and then could explore biological systems and other systems without having to cool to low temperatures. So to just make sure it's clear in your mind how all these experiments are done now, we basically pump from a ground state to an excited state with a laser beam, and after relaxation, we collect these emitted photons. But you can do it on any kind of microscope you like, essentially. It can be wide field, or it can be confocal, can be total internal reflection, and so on. Usually, what we're really observing is the light coming from these small fluorescent molecules like rhodamine or Psi3 or even the fluorescent proteins. And you want to think of these experiments mostly as attaching this as a label to the protein of interest. Here I'm illustrating a cell, for example, but it could be any kind of sample, even in solution. But for a cell, for example, you're wanting to observe the behavior of this protein using the light from the label to tell you where it is. Well, here's a focused laser spot whose size is limited by diffraction, as you've been hearing, Abbe's limit. And so in order to see only a single molecule, it's clear that you must dilute the sample dramatically to make sure that the molecules that are fluorescent are spaced apart by more than the size of this laser spot. If you do that, of course, you see the light mostly from this single molecule if you do the experiment correctly. And there's so much to be learned just by watching single molecules. I want to illustrate a couple of examples. So one thing you can do just by watching individual molecules is to see them behaving in fascinating ways. Here's single molecules in the membrane, the plasma membrane of a live Chinese hamster ovary cell. And those molecules at room temperature are doing this fantastic dance. This is just normal diffusion on the surface of our cells. It's going on in our cells all the time, even faster because we're at 37 C. This was done at room temperature. You also see other things going on in this movie. The molecules turn off. They don't emit forever. They give you maybe 1 million photons and then they give up. You also maybe saw that some of them might be turning back on. But that's mostly observing the diffusion process and learning things about the membrane. Here's another thing you can do with the single molecules. Here's a bacterial cell called Colobacter crescentus, and I want you to just follow this time-lapse movie. Now we're looking over a very long time scale of one molecule labeled with yellow fluorescent protein, a molecule called MREB, and there you see that molecule is doing something completely different. Not diffusion like you saw up here, but moving in a line across the cell, and then turning and then moving in a line coming back the other direction. So line across, a line there. That is not diffusion. Diffusion is random. It keeps changing direction all the time. This is linear motion. Something else is going on. The molecule is telling us, in this case, that it's being moved along by the formation of the cell wall. The MREB is involved in the synthesis of the cell wall. And it's the light and the motion of the molecule that tells you what's going on. Here's a materials example. Completely different, solid, room temperature. It's again a crystal of paraterphenyl, but watch this movie. You see it's completely different than the others. There are a bunch of molecules that are stable and that are fixed in this crystal and don't move around. And then there's a bunch of others that look like little flies in the night sky, fireflies, if you have seen them before, that they are moving around and they're moving in sort of directions, strange directions, mostly up, and mostly horizontal. They're moving in the cracks of the crystal. The motion here of the molecule is telling you where the defects are in this crystal. So there's applications in biology and material science in many, many areas just from watching single molecules. But that alone is not super resolution. So let's start talking more about super resolution. And Stefan Hell described this beautiful idea that he had about stimulated emission depletion and all sorts of other relatives of that approach, where you use a pattern of light to control where the molecule is. But how can we use these single molecule labels to surpass the Abbe limit? So the answer to that is very simple. First, we have to image single molecules, which I've described to you. And then you have to add to that 2 key ideas. So let's talk about the first key idea, which I like to call super localization. A way to think about this, by analogy, is to recognize that suppose we wanted to find the position of this cinder cone. It's a beautiful cinder cone in the middle of Crater Lake, Oregon, which is a volcanic lake. And if you have a chance to go there, I urge you to do so. But this little cinder cone, we have a scale bar here. This is 120 times 10 to the 9th nanometers. Okay, so it's human scale, right? And you know that if you take your little camera and your cell phone and walk up to the peak of this mountain, you can read off the coordinates of the peak of the mountain to great accuracy. Well, that's what we want to do with the molecules. Find the position of the molecules by fitting the shape of the single molecule image. Here are some of these single molecule images where I'm just showing you in black and white, but the important thing is you can blow up the image from the molecule and spread it over many pixels of the detector of your camera. You can see the pixels here. In cross-section, here's those pixels, and notice they have different numbers of photons in the different pixels. And this is a shape, a characteristic shape, that's coming from the microscope. It's called the Airy function or a Gaussian function. And you can fit it with a function, a mathematical function that has as its parameters the width of the function, which is, of course, diffraction limited. But the other parameter is the center position, the location of where the Gaussian is. And this center position, you know to much, whoa, what happened here? This center position, you can know to much better accuracy, precision than the width. In particular, your estimates of the center position have a distribution like that. This is the error distribution for the location of the molecule. That has a width sigma, and sigma is given by the Abbe limit, the diffraction limit, divided by the square root of the number of photons you detect. So if you detect more and more photons, let's say 1 million photons, then you can learn the position of the molecule to, let's say, 20 nanometers. You detect more photons than that, and you can do even better. The idea here is that every photon that lands on the detector is an estimate of the position of the molecule, but to get a good estimate, you have to collect a certain number of photons. Okay, so that's the idea for localizing individual molecules that are separated apart. And this super localization has been used in biological imaging for some time, in particular looking at large objects but finding the centroid. But that's not quite as important for us. It's much better to find the positions of single fluorophores, because the fluorophores are just a few nanometers in size. And this was done in a couple of experiments over time, and many, many experiments not listed here. The general idea is that if all the photons come from the same nanometer-sized object, you can extract its position to high precision. Here's a little, if you like, simulation that shows you how we do our experiments. Here's the single molecule, but it's moving around, and you can see it on the camera there. And then for every camera frame, you fit the position. The green, again, is the error of your estimate of the position. And you finally plot positions as a function of time, showing you how the molecule moves around. So that's great. But it's not super resolution. It's one object, and it is not really distinguishing two objects that are close together. So there were a number of important steps that moved forward along the lines of distinguishing objects, and Eric wrote an important paper in 1995 saying that you could use some additional control variable to separate these overlapping molecules in another dimension. And spectral tunability was proposed, and then spectral tunability was used to achieve super resolution in 1998, but only for about 7 molecules. Other people realized that you could use fluorescence lifetime maybe, or photo bleaching, or colors, or blinking to try to get some super resolution. But these early demonstrations didn't really solve the problem for a huge number of molecules. So that's where the big idea, number 2, comes in, a great idea of Eric Betzig's. And the key idea here, since you can't read it, it says active control of emitter concentration and sequential imaging. So let me illustrate the idea. You want to see this structure. And so you've got your labels decorating that structure, your fluorescent labels. The problem is if you let them all emit at the same time, then all of these images overlap and you just see a blurry fuzz that's coming from the fundamental diffraction limit. So to bypass diffraction, you just make sure they're not all on at the same time. You, as an experimenter, need to actively control the emitting concentration. Suppose you use photo activation, then only a few would be on at a time if you activate them very, very weakly. And you can find their positions then by super localization, use them up, photo bleach, turn on a few more, use them up and photo bleach, and in the end you'll get a whole lot of positions by this time sequential sampling that you can put together in a pointillist reconstruction of the underlying structure. So that's the idea, and you get detailed far beyond the diffraction limit by doing this. And I heard about this idea for the first time in April 2006 from Eric, describing the work that he was doing with Harold Hess, and he called it PALM. And you'll hear more about that probably a little bit in a moment from him, but very quickly after that, Storm from Xiaowei Zhuang, F-PALM from Sam Hess, and then Paint from Hochstrasser, and all these other groups jumped in and said, hey, I know of a way that I can control the concentration of the molecules and implement this beautiful idea. At this time, we decided to do something called YFP reactivation, and of course we didn't use an acronym for it, so it was totally forgotten, right? You know, you got to have those acronyms. As you see, you know, Stefan made a new acronym, 3 different acronyms. So to try to rectify this a little bit, just for fun, I'll add one more acronym to this big pile that's mechanism independent, single molecule active control microscopy, or SMACM. Okay, but let's go back and look at this YFP reactivation in a little more detail, because where did that really come from? Well, when I heard about this PALM idea, I said, well, what can we do to illustrate this in another way? And I remembered an experiment that was done with my postdoc back in Rob Dixon, back in 1997, also in collaboration with Roger, because his postdoc sent us some material, some of this particular yellow fluorescent protein with a few mutations to make it absorb at a longer wavelength and emit at a longer wavelength than GFP. And indeed, we could image single copies of YFP. So that was an important step, of course. But that isn't all we saw. As before, when you start seeing a new regime, a whole regime that hadn't been explored, what happens when you observe a single YFP? You see new effects. Blinking, for example. Here's the molecule emitting, 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 and then it's off for some time, and then back on, and then off and back on. That means that the molecule is cycling and giving us photons, but sometimes going into a dark state, probably an isomerization of the chromophore in YFP. It can return and start emitting again. He also saw that sometimes it would go into a long-lived dark state and be completely off, over here in this energy diagram, but a little bit of blue light can restore it back so it can start emitting once more. That's sort of this photo-induced recovery of YFP. And so you can see here now there's blinking and photo control available in fluorescent proteins as well. And after this, a number of people optimized this effect for switching properties, photoactivation properties, and so forth. Those proteins are now available in many different colors. Let's see what the data look like. Here's some bacteria that I'm showing in white light imaging right here. And here's the movie. The movie shows an incredible blinking process. And that is the blinking that we used for super resolution imaging. In every frame of this movie, you find the locations of the molecules and write down and then look back and see what the underlying structure is. You can apply that to different proteins in bacteria, because the problem is bacteria are pretty small. They're only a few microns in length and 500 nanometers across. Here's 3 different proteins labeled with YFP, and here's the super resolution images. So this, of course, is what makes us all so excited. If you had this before and could not see, now you see this. And it just means that there's a great deal of exciting new effects available to be explored in cell biology when you can look far beyond the diffraction limit. You can use that paint idea from Robin Hochstrasser, which means that there are molecules that are diffusing in solution which are dark, but when they bind to the surface of something, they become bright. We demonstrated that using this molecule, saxitoxin, synthesized by Justin Dubois, and where the saxitoxin has been labeled with a fluorophore. There's a cell up here, a really huge cell off screen, which is a PC12 cell, a neuronal cell, a model cell. And here's one of the projections from that cell that is very much like an axon, and it's being lit up by this molecule binding to voltage-gated sodium channels on the surface of the cell. Live cell, and we could even take this time-dependent image, where I hope you can see, even though there's various lights interfering, I hope you can see that there are projections that extend out over time. This is with 500 millisecond time interval. There's another one, and so forth. These sub-diffraction structures are dynamically growing and retracting, and they can be observed by this sort of target-specific paint. Well, let me just, since time is short, quickly sort of say that really the excitement now for the new work in this area is to come up with new chromophores. Here's one, I won't describe it, but it's a molecule that you can turn on with light, and you can attach it to the surface of bacteria just by simple chemistry, and then see all of these structures that are sub-diffraction structures, these stalks. You can use interesting optical tricks to get 3-dimensional imaging, and these optical tricks allow you to get 3-dimensional images of these bacterial cells that I'm showing here very beautifully. This is the surface of the bacterial cell in this particular case. So overall, so many things have happened in the single molecule area and the super resolution area that this whole area of science is impacting not only chemistry but also physics and of course biology, and working at this limit has produced fantastic contributions by people all over the world. There's no time for me to properly acknowledge everyone, so if you watch really quickly, you'll see there's the previous lab members, the current lab members, and we of course call ourselves the guacamole team because one molecule is of course one guacamole. It's i.e. 1 over avocado's number of moles. So we have to apologize to Amadeo Avogadro for producing this very bad joke. We have this no ensemble averaging logo, and collaborators, colleagues, mentors, funding sources, family, and all these people. And of course, thank you for your attention.